Hello, I'm Lisa Tucker-Kellogg at the Duke-NUS Medical School in Singapore. Today I'm presenting simulations of cancer evolution looking to predict which drug-resistant phenotypes will arise under synergistic versus non-synergistic drug combinations. I have nothing to disclose. A popular strategy for defending against future development of drug resistance is to combine drugs giving a combination therapy, particularly if the drugs have synergistic efficacy, meaning greater than additive effect. However, if one of the cancer cells develops single drug resistance, then having a synergistic therapy would give that cell a greater reward for developing single drug resistance compared with the size of reward under an additive therapy. The larger reward means a larger fitness advantage at grabbing resources compared with neighbor cells that may be dying off. This perverse reward is called the paradox of synergism, and our previous work showed that, unfortunately, there are conditions where this effect can accelerate drug resistance, can cause drug resistance to develop faster compared with additive drug combinations. What we don't know yet is what the paradox of synergism means for the type of drug resistance likely to arise for cancer phenotypes that have partial drug resistance or mixed levels of resistance against the two drugs. In this work, we perform computational modeling of partially resistant phenotypes to estimate the growth rates for each phenotype of cancer cell and to simulate how the population evolves over time. Results show, first of all, that a lot of simulations are not very interesting, either because the cancer is too strong for the drugs. For example, if the mutation rate is too high, in which case all effective combinations will quickly become ineffective, or else because the cancer is too weak for the drugs, in which case any decently effective combination can totally eradicate the disease. So there's only an intermediate range of parameters and scenarios where it even matters how we choose among the different possible types of combination therapy, where the long-term outcome depends on our choices. The interesting result we found can be summed up like this: the fitness advantage of resisting both drugs equally versus resisting one drug more than the other depends on whether the drugs are synergistic or not. Now let me explain what that means. If we consider the theoretical landscape of all possible modifications, mutations, and gene expression changes that can allow a cancer cell on the left to transition from being fully sensitive, that's 100% sensitive to drug A, 100% sensitive and responsive to drug B, into a state where it is fully resistant on the right, zero response to drug A and zero response to drug B. And if we use terminology where a partially resistant intermediate state that's equally resistant to both drugs in green is called symmetric, and where states that are more resistant to one drug than the other, above and below in blue, we call those asymmetric resistance, then our finding is that synergistic combination therapies tend to drive cancer cells toward the blue corners and away from the green paths. After a combination therapy that is synergistic, the resistant cells tend to be in asymmetric states and moving along asymmetric paths, and the blue phenotype is enriched in the tumor cell population. Rather than giving a lot of math, what I want to tell you is that there is always some enrichment, regardless which parameters we assume, but the amount of enrichment, the quantitative effect, depends on the exact mathematical assumptions and the context we're modeling. In summary, cells escaping a synergistic treatment are more likely than random to develop asymmetric toward one drug more than the other. That means one drug would become ineffective and the other might still be useful for treatment. In contrast, cells escaping antagonistic combination therapies would be more likely to develop resistance along the symmetrical paths in green. I hope this short talk has given you a taste of how theoretical modeling can provide testable predictions that complement the kinds of hypotheses we tend to get from experimental work. And I hope you'll contact me if you'd like model-based predictions for your cancer evolution in your context. Alicia Saputra is the first author of this work, and I'm grateful to the whole team. I'm grateful to funding agencies for supporting this work, and grateful to you for your attention.